All right, welcome back everyone. So we're going to pick up with this new part now, looking at um, our first main macronutrient, our first kind of large scale nutrient, and this is going to be our carbohydrate, right? So carbohydrates are essentially think sugars, right? So think your various sugars, and these carbohydrates are going to exist in various forms, okay, various forms. So the first two that we'll think of, and we'll kind of come back to these in a little bit, especially whenever we do carbohydrate metabolism, are what are called mono and disaccharides, right? So a monosaccharide and a disaccharide. So to understand these, simply think of the root of these words. So the prefix mono means one, and the prefix di means two. And then that ending saccharide means sugar. Okay, so monosaccharide just simply means one sugar or one sugar unit, and disaccharide means two sugar units, right? So things like glucose, fructose, galactose, these are all examples of monosaccharides. You can see a single individual sugar unit. It's got carbons, it has hydrogen, so each of these points here, each of these corners is a carbon. It's got hydroxyl groups, these OH groups here, these hydroxyl groups. And the difference between the glucose, the fructose, the galactose are just slightly different arrangements of some of these carbons, right? So instead of having six, maybe there's five. Maybe it's arranged in a slightly different orientation. But the idea here is that it's a single unit, right? You put two of these units together and you get what's called a disaccharide. So over to the right, you can see two units bound together, two individual monosaccharides bound together to form a disaccharide, right? So glucose and galactose, you get lactose. We'll come back to lactose in a little bit. You add glucose and fructose, and you get sucrose, right? There's a third one called maltose, right? So all of these are two individual monosaccharides that have come together to form one single disaccharide, right? So one unit versus two units, right? Now, as we'll talk about in a little bit, in order for us to absorb and use our carbohydrates, our sugars, they have to be in the monosaccharide form, right? So we've got to get our disaccharides, and then this other group called polysaccharides, we have to get them into a single unit form, right? So especially here in the case of polysaccharides, right? So that prefix poly means many, and so this is many sugars, right? Many sugar units. And so for us, right, as humans, as animals, we're going to talk about glycogen, right? So glycogen is sometimes called animal starch, right? So a polysaccharide, which is essentially just a bunch of individual glucose molecules that are strung together, right? Strung together. So think bread, think potatoes, certain types of grains, beans, peas, things like that. Those are all examples of polysaccharides, right? So those are starch. You can also have fiber. Fiber, however, is so if you look here, this cellulose fiber, it's indigestible, right? So we can't chemically break it down, so therefore we can't absorb it, right? However, fiber does still have various health benefits, right? And so there's two types of fiber, one called soluble, one called insoluble. The idea here with the fiber, though, is that it's passing through our gut largely unmodified, so it's not being digested, therefore it's not being absorbed. It can add bulk to our feces, so it can aid in the elimination of waste product, right? Waste material in our gut, in our digestive system. It can also help to slow down the absorption of glucose, so preventing major, glucose spikes after eating, things like that. And then there's also evidence that it helps lower, it may lower risk of cancer. It can help to kind of draw potential toxins and whatnot out of the body with this fiber as it's passing through the gut. Of course, largely unmodified and preventing the absorption of these potentially dangerous toxins, right? Also, cholesterol as well, right? So it can help lower blood cholesterol levels by preventing the absorption of cholesterol, right? Or preventing as much cholesterol from being absorbed, right? So carbs, lots of different functions. Again, they're going to be converted down into individual monosaccharides, so think glucose, right? And again, as we will see in future lectures, when we do carbohydrate metabolism and we do talk about cellular respiration, we'll talk about how we break down glucose. So we break down, glycogen, which is a starch. We break that down into glucose, and then in cellular respiration, we're taking that individual glucose and we're breaking it down even further, right? Even further. And so we're going to break it down. We're going to remove the carbons. This is why you breathe out carbon dioxide, right? And ultimately make ATP. Okay, so that's what we're kind of getting to eventually from our carbs and from our individual monosaccharides as well, right? So when we're digesting carbs, again, we have to break them down from their more complex structure. So meaning the polysaccharide or the disaccharide form down into the individual monosaccharide form, right? And then once you break that down into the monosaccharide form, now you can begin absorbing it, right? So this here is showing an intestinal cell, right? This is the lumen, so this would be the inside of the, say the small intestine. Here's our epithelial cells, our submucosa, then there'd be blood supply over here, right? That it's that glucose is moving into, right? So we have different types of glucose transporters. This GLUT, these stand for glucose transporter. Sometimes they're just referred to as GLUT. So a GLUT1, GLUT5, so on. These are glucose transporters either on this apical surface or on this basal surface of the cell, and it's moving the glucose or the fructose or whatever other monosaccharide it is into the intestinal cell, and then moving that out of the cell, ultimately into the bloodstream, right? So again, before you do that, you have to break it down. So we as individuals, we typically need somewhere around maybe 130 grams of carbs a day. Again, these numbers can vary depending on lots of different factors: nutritional status, size, activity, things like that. But it's typically less than a quarter of our calories should come from sugars, should come from carbs, right? So how do we get our carbs? How do we get those polysaccharides and disaccharides into these individual monosaccharide forms, right? Well, you have to digest it, right? So this is going to be chemical digestion, right? Chemical digestion. And so we are going to use an enzyme called an amylase. Amylase enzymes are those that break down carbohydrates, right? So from their polysaccharide form or their disaccharide form into their monosaccharide form. These can exist in two different places, one from the saliva and from the pancreas as well. So if it's from the saliva, it's from our salivary glands, it's salivary amylase. This is beginning carbohydrate digestion in the mouth, right? So the breakdown, the chemical breakdown, chemical digestion of carbohydrates begins in the oral cavity, right? We take that food that we consume, that carbohydrate that we consumed, we swallow it. So think back to the phases of swallowing that you've talked about when you did digestive system. We swallow it, and that moves into the stomach, and of course in the stomach there's lots of acid, right? And so what's going to happen to that enzyme is that enzyme is going to become denatured, right? That amylase is going to be destroyed. So we're no longer going to be chemically digesting carbohydrates in the stomach with that same salivary amylase, right? Because that salivary amylase is now being destroyed, right? So however, we may not have completely and totally chemically digested the carbs. And so when you move out of the stomach, that chyme that's moving out of the stomach into the small intestine, right, into the duodenum, you get an additional addition. That's good. You get an additional amount of amylase, and this is called pancreatic amylase, right? So the secretions of the pancreas, included in those secretions is more amylase, right? And so you need that because that salivary amylase that was from the saliva in the oral cavity, it gets broken down in the stomach because of the very low pH. And so now this addition of the pancreatic amylase is going to continue the digestion of the carbohydrate further into the individual monosaccharides, and then as we saw on the last slide, then you can begin actually absorbing those individual sugars. Okay, so you break them down from the polysaccharide form to disaccharide form, ultimately to the monosaccharide form. You absorb it, and now you've got this glucose in your blood, right? So we're going to have changes in our blood glucose level, right? So how do you regulate that, right? Of course, after you eat, there's going to be, and this could be after you eat something sugary or drink something sugary, a carbohydrate meal or drink mixture, you're going to have changes, elevations, that is, after you consume the carbs, you're going to have changes in your blood sugar, right? And so think back to our endocrine system discussions, right? You're going to use the products, the hormones from the pancreas, right? So that's going to be your hormones insulin and glucagon, right? So recall insulin is going to help lower blood sugar, right? So in response to an elevated blood sugar, right? So here's our stimulus. Blood glucose level rise, it increases, right? Those levels rise. We're going to release from the beta cells. Remember, beta cells produce insulin, alpha cells produce glucagon. Beta cells release insulin. Insulin stimulates the uptake of glucose, right? So just like we mentioned again a few slides ago, into that intestinal cell, perhaps into the liver, it's going to take it out, not into the intestinal cell, but into our somatic cells, right? Just so our body's tissues. They will take up glucose and thus lower blood glucose levels until they fall back down, and then maybe they fall down too low, and now we have low blood sugar. We're at a blood sugar of, I don't know, 60 or 65 or 70, right? Whatever the number is. And now we're going to do the opposite. So now we're going to release glucagon from alpha cells, and the exact opposite is going to occur, right? So we're going to release glucose. We're going to break down glycogen. Glycogen, again, is think like animal starch. It's a polysaccharide. And then we're going to use that newly released glucose to elevate our blood glucose levels, right? So we keep it in this very tightly controlled range, right? 90 to 100, 90 to 110, right? Kind of whatever reference you're looking at, it'll vary slightly. But we're trying to keep it within this nice narrow range, right? Now, of course, you can have alterations in this system, right? So think diabetes. You can have type 1 diabetes in which the pancreas does not produce the insulin. You can have type 2 diabetes in which you are not responding to the insulin, right? So think like insulin insensitivity, typically associated with obesity, weight gain, things like that. And then just the increased blood sugar itself can cause damage to blood vessels, right? And so these can be blood vessels in the eye. These can be blood vessels in our kidney. So you can get something called diabetic nephropathy. You can get diabetic retinopathy, which is affecting the blood vessels in the eye, so you get vision issues. You can have neuropathy, so issues with sensation in the digits, so think maybe like feet and hands, all due to this prolonged exposure of the blood vessels to blood sugar, right? So transient changes in blood sugar, after you eat or you haven't eaten, that's normal, right? It's going to rise, it's going to fall. It's when it's more consistent. So you have consistently high blood sugar levels, right? Or you have really low blood sugar levels, which can be extremely dangerous, right? And so there are tests in the clinical setting that can kind of gauge your overall blood sugar levels over a period of time. This is called your hemoglobin A1C test, sometimes just abbreviated your HbA1c. And this is essentially a measure of red blood cells exposure to glucose over a certain period of time. Red blood cells live, about 120 days or so think 4 months. And so you can measure this exposure of red blood cells to glucose, and you can get a good idea of how well blood sugar has been regulated, how well it's been maintained over a certain period of time. Okay, so that's a little clinical correlate. Another clinical correlate is lactose intolerance, right? So this is something that you may be lactose intolerant. You almost certainly know somebody who is lactose intolerant, right? And even if you don't, we can still talk about what it is. So this is also sometimes called lactase non-persistence. So anytime you see this ending ose, O-S-E, think sugar. Anytime you see ACE, A-S-E, think an enzyme, right? So lactose intolerance means you can't tolerate lactose. The other name for this is lactase non-persistence, meaning you no longer produce lactase. Lactase is the enzyme that breaks down lactose, right? So we're called lactose. It is a disaccharide, right? So we need to break down that disaccharide into a monosaccharide, right? And then we can absorb those individual monosaccharides. So individuals who are lactose intolerant, they're unable to consume products that have lactose, right? So think dairy, because they don't have the enzyme to break down the lactose. So it remains in the GI tract undigested, right? Not chemically broken down, and this results in the cramping, diarrhea, bloating, abdominal distension, all of the GI complications that come with consuming dairy or lactose if you're lactose intolerant, right? So if you can drink milk, if you can eat ice cream, if you can consume anything with lactose, dairy, and not have complications, right, then that means you have the enzyme, right? So what happens normally is as mammals, right, we drink milk as newborns, as infants, and in most mammals that stops after a period of time, right? So after infancy, in most mammals, you no longer the individual, the offspring, no longer consumes the milk of its mother, right? That's the kind of we'll call it normal, right? So the reason for that is there's a gene, there's a region of DNA, a gene that codes for the lactase enzyme, right? And after a certain period of time, after infancy, that gene turns off. So when that gene turns off, you no longer produce the enzyme, so you're not doing transcription, you're not doing translation, you're not producing the lactase enzyme, so you can't break down lactose. So you're lactose intolerant, right? And we'll call that the normal condition. Okay, we'll call that the normal condition. That's what's quote unquote supposed to happen, right? If you can drink milk, then you have a mutation in your DNA that keeps that gene switched on. So in other words, you continue to produce the lactase, so you're able to continue to break down the lactose. So you can drink your milk and eat your ice cream and cheese and everything else that has lactose in it. You're able to still consume that without any GI complications, right? Because that gene has a mutation in it that keeps it turned on, if you will, and so you continue to produce that enzyme, right? So why does this happen? Well, we can think of it from an evolutionary perspective, right? And so when domesticating animals and domesticating cows became more common, there was an advantage, right? A selection advantage to having a condition that allows you to continue to consume milk, right? And so there was an article published by NPR a while back that was talking about kind of the evolutionary background, the evolutionary foundation of lactose intolerance, and they were discussing the role of famine, and this was some maybe 8,000 years ago or so when kind of some of these animals were starting to become domesticated. If there was a famine and nutrients and resources and food was scarce, then if you had some mutation that allowed you to continue to drink milk of the domesticated cows that you have, then you're going to have a survival advantage, right? And so if you survive and you have that gene and it happens to get passed on to your offspring, then that gene is going to kind of persist and it's going to continue. Hence why this condition is also called lactase non-persistence, right? So if you have this mutation and you now have this survival advantage over individuals who can't consume the milk, so they're more likely to be famine, they don't have enough nutrients and resources, or they try to drink it and they have diarrhea and they get sick and they die. Now you have this advantage, and so this can be passed on if this is occurring in a sex cell in which it can be passed on during reproduction. Then this mutation is going to persist, right? So it's kind of following just a typical evolutionary explanation, and it makes really good sense when you consider it from that perspective, right? So in modern times, right, if you're lactose intolerant, and if you are, you know this, you can still drink your milk and eat your cheese and ice cream and whatnot. You just have to provide yourself with the enzyme, right? You have to take lactase, or the medication is called Lactaid, right? And you can just get it over the counter. You can usually take the pill, the Lactaid, which is the lactase enzyme, with your first bite of your meal, and you're now able to break down the lactose into the monosaccharides. You can absorb it, and you can enjoy your food, right? So let's stop here, and we'll pick up in our next part, and we'll begin looking at lipids.